Hello, good afternoon. My name is Dr. Lorna Lancaster, and I have with me here Dr. Ian Hislop. And we are from the School of Pharmacy at the University of Lincoln. And today we are going to be talking about microbial warfare. And in particular, we're going to be looking at antibiotic resistance or antimicrobial resistance, but focusing on antibiotic resistance. And so we're going to be focusing on bacteria within this session. And the title of our talk is "The Fight is On," because we are at war with microbes because we are running out of different ways of killing them off that we have at the moment. So just to look at what are microbes, first of all. So in the field of microbiology, there are different areas that we look at when it comes to microbiology. We have the world of parasites, and these are multicellular or single-celled eukaryotic organisms, and they require a host for certain parts of their life cycle. And they require the host for either reproduction or for certain nutrients. But some examples include worms or head lice. There's also fungi, and we're not talking about the mushrooms that kind of live in the forest. These can cause an infection. They can be multicellular or single-celled eukaryotic organisms. And the means of areas, they obtain food from other organisms, which makes them fungi, and they can cause infections such as thrush. Bacteria, which we're going to be focusing on in particular today, are single-celled prokaryotic organisms, and these can be found anywhere on the planet as long as there is some form of water. So name an environment within the planet, and you would be able to find bacteria there of some kind. So even in the coldest environments at the bottom of the Arctic, or in the hottest environments at the bottom of the ocean near hot springs, you can find a form of bacteria. In microbiology as well, there is also the most minuscule, which is viruses, and we are in an environment that everybody knows the word virus, I think. And viruses are acellular. They have the absolute minimum requirements for them is a capsid, so a protein coating, and some nucleic acid, either DNA or RNA, not both, just DNA or RNA. And they only replicate using the host cell. They cannot reproduce on their own. They have to use the tools from the host cell. So we're then going to look at what are antimicrobials. These are substances produced by a microorganism that in low concentration inhibits the growth of another microbe. The majority of antibiotics are based on naturally occurring compounds. So we had these compounds originally or found them originally in another microorganism. So the first one that we found was by Alexander Fleming, who found an antibiotic attacking Staphylococcus aureus. And basically, he wasn't a particularly tidy scientist. So he went away on holiday. He was growing Staphylococcus aureus, and he was trying to find something to kill it off. But he went away on holiday and left his plates on the bench. Very, very naughty scientist. And he went off to on his holidays, came back, and he had a contamination. But what he noticed was that contamination was killing off the Staphylococcus aureus, the bacteria on the plate that was killing them off. So from then, that was the first penicillin. From then, we have found many other different antibiotics, and we have also altered the antibiotics that we have to make them stronger, better, more effective, less antibiotic resistance capable. And most of the ones that we have now are either semi-synthetic or synthetic, so we make them, or we purify them from the environment and then change them slightly to make them better, as I said before. Okay. So what we need to think about when we're talking about antimicrobials is how we get them to kill off the bacteria and or the other microorganisms that we have to be able to fight when it comes to infection. And the key is selective toxicity. So we have to be able to find something about the microorganism that is different to human cells. So we have to think about how we can target the microorganisms that come to us and infect us. So we have antibacterial drugs, which we actually have most of. We have the largest number and diversity of antimicrobial agents within that are against bacteria, because these are actually very different from us. They have prokaryotic cells, which means that they have different cell walls, they have different ribosomes, they have different membranes. So we can actually find a lot of their cell to target. So it means that we can target them and not damage the host. We have fewer drugs for eukaryotic infections like fungi and parasites because they are eukaryotic, just like our cells, so the human host cells. So we have less targets within their cells that we can use to destroy them. So we have to think about what we can use in those cases, and there are some differences because they are different organisms, but there are less of them compared to bacteria. And there's even fewer antiviral drugs because there's actually very little to a virus to be able to target. So we have found it very difficult to find antiviral drugs because a lot of the time, all of the time, the viruses are using the host cell to be able to replicate. So you would be targeting the host cell in a lot of cases. There are a few, but not as many compared to antibiotics. So we're going to be focusing on antibiotics. Okay. Next slide. I think. There we go. So how do antimicrobials work? So they must bind or interfere with an essential target. So this is talking about this of selectivity. So it has to bind to an essential target. So that is something that has to be there for the cell to be able to survive. So a lot of the targets can be the cell wall, which keeps the bacterial cell intact. Or it has to be a metabolic pathway of some kind, which allows the bacteria to grow or divide. And it has to be something that either stops the growth of the cell or it has to cause irreparable damage to the cell. And for the example of penicillin, penicillin attacks the cell wall of the bacteria, and the penicillin stops the different building blocks of the cell wall from joining together. So in a growing cell, what happens is the hole becomes bigger and bigger and bigger because the cell wall doesn't attach itself to each other. And then because of osmosis, the water comes into the cell and this cell lyses. But this only affects growing cells. So it doesn't necessarily cause irreparable damage to cells that aren't growing, but it does to those that are growing. So oh, come on. Next slide. Slides are being a bit slow. Oh, there we go. So what was life like before antibiotics? So before antibiotics, there was a lot of disease that we couldn't treat. And unfortunately, there was a lot of death from infections that we now kind of accept that people are fairly unwell from, but will survive. So just as an example, some of the images here, we have bubonic plague. And there was huge pandemics a number of 100 years ago. And this was caused by a bacteria that traveled within rats. Back then they didn't have antibiotics, and millions of people died from bubonic plague. And it's now easily treatable so we still have some cases of it, and we treat with antibiotics. And we don't have the pandemics that have occurred previously. Also, if there was any operations before in their life before antibiotics, a lot of the time people wouldn't survive these operations when they're required, as an infection could set in, and this would cause or could cause gangrene. And other sort of similar infections, and then there would be further amputation required, and there would be further disease, and often people wouldn't survive the infections that would set in after the operation. And in a lot of cases, for example, in World War I, many soldiers died of bacterial infection from gangrene rather than on the battlefield itself. And then we have what we now see as fairly straightforward infections that can be treated very easily. Impetigo, which before the world of antibiotics, these wouldn't be treated, and a lot of the time left quite significant scarring and fairly horrendous sort of disease profiles. So we're lucky to have antibiotics now, as we don't see nearly as many of these infections. Okay, so we have antibiotics now, but unfortunately we're actually getting to a stage that we may not be able to use them in the way that we have been doing in recent years. And we may end up in a situation that is similar to what it was like previous to antibiotics being discovered. And that's because of antibiotic resistance. So what is antibiotic resistance? It is the inability to kill or inhibit the organism with clinically achievable drug concentrations. And bacteria can have this as an innate ability. So this is a naturally resistant bacteria. So they either don't have the target that the antibiotic can bind onto and attack, or they have a barrier for the antibiotic to get to that antibiotic. The bacteria, and that this is what we are now having to fight, is the resistance that is acquired. So as we expose bacteria to antibiotics, they like to change. So they have started, or way back when, they have mutated potentially when they've been exposed to antibiotics and they mutate in such a way that makes them resistant to that antibiotic. So they can change the target site, or they can create a defense mechanism so that the antibiotic cannot reach the target site to allow killing of the cell. So that can be a direct mutation, or they can acquire DNA from another bacteria that's in the surrounding environment. So bacteria like to share their genetics, and they pass them on to each other when they're living in an environment. So if you have one bacteria that's resistant genetically to an antibiotic, they like to pass that on to other bacteria in the surrounding environment, and they end up passing on that antibiotic resistance. So what's caused antibiotic resistance? So it can be overuse or misuse of antibiotics. It can be inadequate levels of antibiotics at the site of infection. It could be the duration of the treatment being too short. Or it can be overwhelming numbers of organisms. So most of these situations, it means that bacteria are observing antibiotics or seeing antibiotics and it being able to adapt, but without being killed off by the antibiotics at such concentrations. So if you're given antibiotics when you have a virus, it means that the bacteria that are living naturally in your body are seeing antibiotics that maybe that aren't killing them off, but they are seeing them and going, we don't like that. So I'm going to change myself to be able to avoid being killed off by that in the future when it's at a higher concentration, which it might be in the future. So we need to be able to ensure that we have antibiotics in our bodies at a concentration where the bacteria are killed off very quickly by it and don't have that opportunity to be able to develop antibiotic resistance. So quite often overuse or misuse of antibiotics is when patients are given antibiotics or they take antibiotics they don't necessarily need for what has been a viral infection, and antibiotics don't work against viruses because they're not selective against viruses. Or it could be that it's the wrong antibiotic for the wrong type of infection. So it means that the bacteria within the environment of the patient are going to be seeing that antibiotic, not being killed off by it necessarily, but they are starting to adapt and mutate to create antibiotic resistance. So some examples of overuse we've started talking about. There can be veterinary practices. It's been banned now in the UK, but there was an instance a long time ago and now still in other countries around the world. So it doesn't happen in the UK and EU anymore, but it does still happen within the world, where antibiotics were given to animals to allow them to grow bigger. So if you take away the possibility of infection while they're growing, it means they can grow more rather than try to fight an infection. So antibiotics were used as to allow the animals to grow bigger to produce more meat. We also provide antibiotics to pets and to farmyard animals. So a dog can't tell you what's wrong with them and why they're feeling poorly. So antibiotics are given a lot of the time to make them feel better. And although that is quite right, it is using antibiotics when it has a potential to be a viral infection. Also within farmyards, if one animal takes an infection, a lot of the time the whole flock or herd are provided with antibiotics. to prevent spread, but that means more antibiotics in the environment and more antibiotics getting into water systems, which means that more bacteria are going to adapt and mutate to become resistant. We've talked a little bit about viral infections. So viruses are quite different to bacteria. They have different target points if you were to have to try and kill them off. So they are not affected by the antibiotics. They don't have the same sort of cell wall or ribosomes. So they can't be killed off by antibiotics. But quite often antibiotics are given with viral infections when GPs or doctors don't know whether it's viral or bacterial. And this means that antibiotics are given, and again, bacteria are exposed to these antibiotics when actually it's not doing any good for the patient at that stage. And we'll talk a little bit later about stewardship. But there is, if we don't control the prescribing and the use of antibiotics, there are more and more antibiotics then in the environment and exposing other bacteria to antibiotics to become resistant. Slide. Change in a minute. There we go. So, what are the consequences of antibiotic resistance? So, we end up with some infections being like pre-antibiotic era. So we have infections which we now take for granted can be treated, will no longer be able to be treated. And there are some infections that no antibiotics that we have in our arsenal can be used against those infections. And there are some people living with antibiotic resistance infections. And there are thousands of deaths each year from antibiotic resistant infections that we cannot treat because we have no antibiotics left to be able to treat them. There are certain species of bacteria that we do have cases. There are no antibiotics left to be able to treat them. And one of these is tuberculosis. So there are certain conditions that we can no longer treat. The treatment of infections is more complex, so a lot of the time we might have to give 2 or 3 different antibiotics to be able to actually treat the infection, and we have to do a lot more testing to make sure we are using the right antibiotic at the time. And if things progress in the way that they are going, things like surgery, where risk of infection is greatly increased, these will no longer be possible without huge risk to the patient, because opening up the body cavity and the risk of infection without it being able to be treated could lead to very significant infections that the patients wouldn't survive. Can antibiotic resistance reduce? Well, there is evidence that reduction of antibiotic use does reduce the levels of antibiotic resistance. And that's because bacteria are living continuously in a world of competition, and they like to adapt to their environment. So we are actually seeing so much antibiotic resistance because with our use of antibiotics, we are pushing them to mutate to survive in a world with antibiotics. But bacteria sort of live in an environment a bit like the game rock, scissors, paper. And when they're living, if you're playing the game with 3 people, generally nobody ever wins, because somebody will be scissors, somebody will be paper, and one person will be rock, and everybody survives. They all have slight advantages. And some a bacteria that is maybe resistant to an antibiotic might have to expend energy to produce an extra enzyme, or they might have to have 2 different enzymes or molecules to be able to take in nutrients, because it's had to block a target site to change, to adapt to an antibiotic or something like that. So a microorganism or a bacteria that doesn't have these mechanisms, when there's no antibiotics, will survive quite well and may outcompete bacteria that do have resistance. So if we were to remove the pressure of antibiotic sort of resistance, so take away the antibiotics, it means that the bacteria would probably get rid of the genetics to keep antibiotic resistance, so that they are no longer outcompeted by the bacteria that are resistant. So in that rock, paper, scissors game, they want to outcompete their competitors within their environment. So by producing an extra enzyme, they are putting themselves at risk of being outcompeted by a bacteria that's not resistant, because they can't take in the nutrients that they need. So there is evidence that antibiotic resistance does reduce, and there's a number of studies, all from quite a while ago, but we have seen that reduced cephalosporin usage is associated with reduced rates of vancomycin-resistant enterococci and methicillin-resistant Staphylococcus aureus. We've seen that decrease in invasive enterococci, methicillin-resistant Staphylococcus aureus, and resistant gram-negatives after improving antibiotic prescribing. And we have reduced cephalosporin usage associated with less Clostridium difficile and MRSA in acute elderly patients. So there is evidence there that if we stop using certain antibiotics, we can reduce antibiotic resistance. So what do we do now, and what are scientists doing now to keep the fight going against antimicrobial resistance and microbial infections? We are looking at epidemiology of antibiotic resistance in the environment, in patients, and seeing where the trends are and what we can change to improve the prescribing practices. We are looking at improving diagnostic tools so that we can tell as soon as possible what the infection is and what antibiotics it's resistant to, so that we can use the best antibiotics straight away to be able to treat the infection without having to maybe trial 3 or 4 methods before we get the true diagnosis. We're also looking at novel methods of treating and preventing infection. So instead of antibiotics, we're looking at other ways that we can use to hopefully prevent infection from happening in the first place, and also being able to treat infections. And the more different weapons we have to fight bacteria in particular, the less likely they are to develop resistance. Ian is going to be talking in a little bit about guidelines on antibiotic usage and stewardship. So I shall pass on to Ian if he wants to have a chat about antimicrobial stewardship.
Great, thanks, Lorna. So as Dr. Lancaster was saying, antimicrobial resistance is now a massive problem worldwide, and we're recognizing that it's a major, it's going to have major impact on healthcare worldwide. And really, we've got two big problems that we have to address. As Dr. Lancaster said, microbes are becoming increasingly resistant resistant to the antibiotics that we already have. And also the other problem that we have is that we have fewer new antimicrobials in the pipeline than we've had for quite some time. Between the Second World War and the 1980s, 1990s, there was a lot of research done on new antibiotics. And with time, a lot of bacteria become resistant to those antibiotics. But since about the 1990s, maybe early 2000s, we've not had as many new antibiotics being, coming on the market than we've had in previous years. And there's been various reasons for that. Perhaps, maybe we've found all the easy targets and we've sort of developed antibiotics against the easy targets on the bacteria. Now we have to look at other targets, and maybe those other targets aren't as easy to target. But also as well, what we've seen is that we've seen a shift in the money that the research companies and the pharmaceutical industry companies have spent on antibiotic research previously. Now there's been a move to spend more money on things like cardiovascular drugs and other drugs, etc. So we've had this lull where we just don't have as many new antibiotics coming on the market, and this has become really, worrying for various governments around the world, because as Lorna explained, we don't want to go back to a pre-antibiotic age where we can't do operations because we don't have good antibiotics to cover them, and we can't treat relatively simple antibiotics. In 2016, the UK government produced a report which has quite a long name. Its proper name is Tackling Drug-Resistant Infections Globally, Final Report and Recommendations of the Review on Antimicrobial Resistance. And it was a group chaired by Jim O'Neill. So most people call it the O'Neill Report. And they looked at ways of trying to correct the problems that we've got with antimicrobial resistance. And they basically sort of looked at ways of reducing that antimicrobial demand by, having global awareness campaigns to, reduce usage or overusage or wasted usage of antimicrobials. And they also looked at ways of trying to improve the number of effective antimicrobials that we've got. So they were looking at governments putting money into research as opposed to just the pharmaceutical industry, relying on the pharmaceutical industry to do the research. They talked about putting the government's money in to develop research as well. So this report was the first major report in the world like this, because it was the first time we'd had a major government globally looking at this problem of antimicrobial resistance and trying to produce a path to get us out of this problem. And now all the other major governments around the world have been looking at this report and producing similar sort of strategies and actions an action plan as well to try and fight this problem. Next slide, Lorna, please. Mm-hmm. I sound like Boris Johnson saying next slide, please. So basically what we've been doing is, globally, is we've started, a process of what we call antimicrobial stewardship. And this is where basically we look at all of the things that we do with antibiotics and how we use them, how we control them, to try and make sure that with time we've got an effective range, we're maintaining an effective range of antibiotics to treat, simple infections through to very severe infections. And so the term antimicrobial stewardship, you may hear this term, it's sort of like an umbrella term to sort of describe all of the actions that we do in our healthcare systems to promote the good use of antimicrobials. And we recognize that this has to be a multidisciplinary approach. Basically, everyone has to be involved. And we have to do measures at different levels. So we have to have, actions and measures occurring at a local level, because we see very different resistance patterns in different communities and different hospitals within our communities. We have to have actions at a national level, with the government promoting the use of, the appropriate use of antibiotics. But we also, as we've seen with COVID and the sort of spread of COVID around the world, we need to have measures at a global level as well. So we've basically got various approaches at these different levels, and a wide range of people involved, from patients through to doctors, pharmacists, other health practitioners, governments, organisations, etc. Next slide, Lorna, please. So this slide just really summarises some of the people, not everyone, but just some of the people who are involved or who have key roles to play in the appropriate use of antibiotics, or good antibiotic usage. At the top there, we've got the prescribers, the doctors, the vets, the dentists who prescribe antibiotics, and increasingly pharmacists also prescribe antibiotics. And it's important that the people who prescribe antibiotics use, basically follow guidelines and follow local guidelines, and also prescribe antibiotics very carefully. They only should really prescribe antibiotics when we know that they're definitely going to be effective to treat that infection. Like Lorna said, some antibiotics, when we're using antibiotics to kill bacteria, they won't be effective in something like a simple viral infection like the common cold or something like that. So we need to use them appropriately. When we use them, we need to make sure that we're prescribing the correct dose for long enough to kill the harmful organism, but not too long and not too short a course. We also need to use what we call narrow spectrum antibiotics that just target the harmful organism and don't damage any of the other microorganisms that live in the person's body. And if possible, try to just use one antibiotic, but with some infections we have to use multiples. On the next slide, I'll talk more about what pharmacists do, but a key group of people now are in the bottom right-hand corner there, the ARMS teams, the antimicrobial stewardship teams. Now in most hospitals and healthcare systems, we have these antimicrobial stewardship teams, which are groups of infectious diseases doctors and doctors, GPs, surgeons, pharmacists, nurses, etc. And they work within the hospital or within that healthcare system to help develop guidelines for antibiotic use, but also to educate prescribers and other people to make sure that the antibiotics are being used appropriately. Nurses are also involved, but also on the previous slide as well, we had all the other bodies like governments, professional bodies and that sort of thing, which also have a role. On this slide, because myself and Lorna, I'm a pharmacist and Lorna's a pharmaceutical scientist, so I guess we're interested in what pharmacists can do to reduce the rate of antimicrobial resistance. Pharmacists are, I guess, the medicine specialists within the healthcare team. So it's our role to make sure that medicines are used appropriately in the healthcare team. And we can do this by making sure that antibiotics are just used for bacterial infections and not viral infections. Some infections, what we call self-limiting infections, like things like sore throats, common colds. These are infections that the body will deal with on their own without antibiotics over, between maybe 5 to 14 days, depending on the infection. So it's important that we don't use antibiotics to treat these infections. We also have a role in preventing, making sure that people are using antibiotics appropriately. And pharmacists also have a role in things like immunization, making sure that people get vaccinated for infections. So some pharmacists are now involved in the COVID vaccination scheme and also minor ailment scheme. Pharmaceutical scientists, I'll maybe let Lorna talk more about what pharmaceutical scientists do, but they have a key role. So Lorna, do you want to comment on what they do?
Sure. So pharmaceutical scientists, they work in designing and formulating new antibiotics. We're also looking at novel ways of sort of fighting bacteria other than antibiotics. So there are different ways that we can treat infection that are not necessarily with antibiotics. So we're looking at vaccines. So vaccines that are developed now are generally for preventative, which is brilliant, because if we can prevent infection before they happen, so you reduce the need for antibiotics, especially prophylactic antibiotics, where you kind of prevent infection by using antibiotics. We can also look at new ways of delivering medications as well, which might reduce the need to have antibiotics in the full body. So it could be new creams or gels that can target specifically the infection in the right place. But pharmaceutical scientists also are looking at making sure that the antimicrobials that we do have are delivered in a high quality and in the best formulation that we can have. So pharmaceutical scientists are involved in sort of development of new medicines that can treat infection, as well as making sure the products that we do have are actually the best way we can deliver them at the moment. So yeah.
So on this slide, like I said, I think everyone has a role to play in antimicrobial stewardship and reducing the rates of antimicrobial resistance. And so these are some of the things that patients or people like yourself can do to try and stop or reduce the development of resistance. The key thing with antibiotics is to follow the advice of the health professional that you're seeing, whether it's a doctor or a pharmacist or a nurse. Follow their advice and only take antibiotics that are prescribed for you, okay? If, say, your auntie, uncle have been given antibiotics to treat their infection, it doesn't mean to say that you should use the antibiotics. So I used to see this quite a lot in the Pacific region, where patients would be given antibiotics, they would go back home and then they would give it to all their relatives. So it's something we only use antibiotics that are prescribed for you. Don't get antibiotics from other sources. Some people have been getting medicines from the internet and from overseas. And sometimes when you go overseas, you can buy antibiotics in things like supermarkets and even in some pharmacies overseas, depending on the country you're going to. It's important that you only use antibiotics, like we said, that are prescribed for you. So don't get antibiotics from other sources. If you're given antibiotics, make sure you take the correct dose at the correct time and make sure you finish the course. And then if you've got unused antibiotics, return them to the pharmacy for destruction. Don't flush them down the drain or the sink or the toilet or put them in the dustbin. Take them to the pharmacy for destruction. And then it's important that you don't share antibiotics with others. So there's some simple rules that everyone can follow, even if they're not a health professional. So back to you, Lorna.
Yeah, so as pharmaceutical scientists, we are looking at different ways that we can target bacteria without using antibiotics. So as we were talking about vaccines, we're looking at new antibiotics, but are they the best way forward? Because resistance does develop, but there is research into new antibiotics. There's research into alternative procedures that can kill bacteria, such as bacteriophage, which is a type of virus which targets bacteria itself. So that kills them off in a kind of natural way, because bacteriophage are everywhere. We can also use antibacterial peptides. So every cell in the world produces peptides that are antibacterial. Can we use these? And you can see some pictures of some of the different things that we're looking at. And there's a lot of research into antimicrobial metals that could be used to prevent infections from happening in the first place. So this is what pharmaceutical scientists and researchers at the university are working on currently. So if you are interested in being a medicines expert, you can study sort of two main subjects. So there's the BSc in pharmaceutical science, and that's study to be experts in the design, formulation, and manufacturing of medicines. Or if you're wanting the sort of more with the patient side of things, so trained to be a pharmacist, to be the medicines expert within the multidisciplinary healthcare team, ensuring the right medicine gets to the right patient at the right time. And that's the kind of difference between a pharmaceutical scientist and a pharmacist. We have open days if you want to visit us. But if you want to keep in touch, we do have a Facebook page at UL Farm, or a Twitter account at UOL Farm as well. So please do come and say hello to us. And thank you for listening.